Welcome to today's deep dive, where we're going to be dissecting a particularly intriguing bacterial genus, Haemophilus.
Ooh, fun.
Now, as medical microbiologists, these tiny troublemakers can pack quite a punch.
They sure can.
And few have a history as storied as H. influenza.
Yeah.
In fact, you might be surprised to learn it was once mistakenly thought to be the cause of influenza itself.
It's amazing how that misnomer stuck even after they figured out it was a virus causing the flu. Talk about a historical footnote that just wouldn't quit.
Right. But for this deep dive, we're laser focused on getting into the nitty-gritty of these bacteria. We're using principles and practice of clinical bacteriology as our guide, specifically the Haemophilus chapter, to build a rock-solid understanding of these organisms.
Excellent choice. That text is a treasure trove of information. And to really grasp how these bacteria operate, we need to start with their classification. Haemophilus, as you know, belongs to the Pastoralaceae family. But it's the individual species that really steal the show.
Yeah, absolutely. H. influenza is the headliner, no doubt. But there are other species lurking in the wings, like H. influenzae, that pique my curiosity. What can you tell us about these lesser-known players?
Well, while they might not be as notorious as H. influenza, they're still quite intriguing. Some, like H. influenza, are considered commensal, hanging out in our respiratory tract without causing much trouble. But every so often they can turn opportunistic, causing infections like endocarditis, particularly in folks with underlying health conditions.
So they're like those quiet neighbors who suddenly throw a wild party.
That's one way to put it. And then you have species like H. ducreyi, the culprit behind chancroid, a sexually transmitted infection we'll delve into later. Each Haemophilus species has its own quirks and clinical significance, which is what makes studying them so fascinating.
Now let's zoom in on the star of the show, H. influenza. What are we looking for when we peer at it under the microscope?
Picture this, a tiny gram-negative coccobacillus. It's a bit finicky, requiring specific nutrients to grow, specifically hemin, known as the X factor, and NAD, known as the V factor.
So that's where chocolate agar comes in. It provides that gourmet buffet they craved.
Exactly. Without those key ingredients, H. influenza just won't cooperate in the lab. Now, what makes H. influenza even more interesting is its population diversity. We have these two distinct groups, capsulate and non-capsulate strains.
I'm particularly intrigued by this distinction. Could you break down the difference between those two groups for us?
Of course. Think of the capsule as a protective shield surrounding the bacterium. The most notorious capsulate strain is type B, or Haib, as it's known. It's the main culprit behind those invasive, life-threatening infections like meningitis, particularly in children. The good news is the Haib vaccine has been a game changer, dramatically reducing the incidence of these serious diseases.
That's a testament to the power of vaccination, for sure. Now, the non-capsulate strains are entitled, don't have that shield, which makes them a bit less menacing, right?
Right. They tend to cause less severe infections, often localized to the respiratory tract, like otitis media and sinusitis. But don't underestimate them. ND can still cause serious problems in vulnerable individuals, especially those with compromised immune systems or chronic respiratory conditions.
So while they might not be as dramatic as Hinba, they're definitely not to be ignored. Now, to understand how these bacteria cause such a range of infections, we need to dive into their arsenal of virulence factors. And if I recall correctly, the capsule is a key player in this story.
Absolutely. The capsule, particularly the type B capsule, is like a master of disguise, helping the bacteria evade our immune system. It's composed of a unique arrangement of sugars called polyribosyl ribotyl phosphate, PRP, which makes it incredibly slippery and difficult for our immune cells to grab onto and destroy.
So it's like a stealth bomber,
Yeah,
evading our body's radar.
a perfect analogy. And it's precisely why encapsulated strains can wreak such havoc. They slip past our defenses, multiply rapidly, and spread throughout the body, leading to those invasive infections that can quickly become life-threatening.
Speaking of weaponry, I'm also curious about lipopolysaccharide, or LPS. It's a component of the bacterial cell wall, but I understand it can be a double-edged sword.
You're right on the money there. LPS is essential for the bacteria's structural integrity, but it's also a potent stimulator of our immune system. It's the lipid A portion of LPS that really triggers the inflammatory cascade. And while some inflammation is necessary for fighting infection, too much can be harmful.
So it's a delicate balance. A little LPS might be beneficial, but too much can trigger a dangerous overreaction from our immune system.
Exactly. In severe cases, this overreaction can lead to septic shock, a life-threatening condition characterized by a dramatic drop in blood pressure and organ failure.
Yeah.
So LPS is a key player in both the bacteria's survival and its ability to cause harm to the host.
Now, what about those pili and other adhesins mentioned in the book? They sound like crucial tools for H. influenza to gain a foothold in our respiratory tract.
You're absolutely right. Think of pili as tiny grappling hooks, allowing the bacteria to latch onto the cells lining our respiratory tract. They're essential for colonization, that initial step in establishing an infection. And it's not just about sticking. Some adhesins also play a role in triggering the release of inflammatory molecules, further contributing to the disease process.
But they're not just passive anchors. They actively participate in the infection process.
Precisely. They're part of a complex interplay between the bacteria and our immune system. And to make matters even more complicated, H. influenza has another trick up its sleeve: phase variation.
Phase variation. That sounds intriguing. Tell me more.
Imagine a master of disguise, constantly changing its appearance to evade detection. That's essentially what H. influenza does with phase variation. It can switch the expression of certain surface proteins on or off, making it harder for our immune system to recognize and target it.
So it's like a chameleon, blending into its surroundings to avoid capture.
A perfect analogy. This ability to change its surface features is one of the reasons why H. influenza can be such a persistent and challenging pathogen to combat.
It sounds like we're dealing with a truly wily adversary. Now I think it's time to shift our focus to the epidemiology of Haemophilus infections. And given the distinct nature of capsulate and non-capsulate strains, I'm guessing their patterns of spread and the populations they affect are quite different.
You're absolutely right. Let's start with Hib, the capsulate type B strain. Before the introduction of the Hib vaccine, it was a major cause of serious illness in young children.
So how was Hib typically spread? And what were the most common diseases it caused?
Hib spreads primarily through respiratory droplets, those tiny particles expelled when we cough or sneeze. Young children, particularly those under 5 years old, were especially vulnerable because their immune systems are still developing. The most feared complication was bacterial meningitis, an inflammation of the membrane surrounding the brain and spinal cord.
Meningitis is a terrifying diagnosis. And I remember reading that Hib was also responsible for other serious infections, like pneumonia and epiglottitis.
You're right. Before the vaccine, Hib was a major public health concern, causing significant morbidity and mortality in young children.
The development of the Hib vaccine was truly a game changer. Now shifting to non-capsulate H. influenza or NSIRE, it seems like its epidemiology is quite different.
Indeed, NSIRE tends to affect older children and adults, and it's more often associated with localized respiratory infections like otitis media sinusitis and bronchitis.
Those are common ailments, but usually not as life-threatening as the infections caused by head abration.
That's generally true. NC head infections are typically less severe, though they can be quite bothersome and occasionally lead to complications, particularly in people with underlying lung diseases or weakened immune systems.
So while NSI might not be as dramatic as Hib, it's certainly a significant player in respiratory illness, especially in certain populations.
Precisely. And to understand how to effectively diagnose and manage these infections, we need to delve into the world of laboratory testing.
That's a great segue. Let's switch gears and explore the fascinating world of laboratory diagnosis. I'm always amazed at the intricate techniques used to identify these microscopic culprits.
It's like detective work, isn't it? We use a combination of traditional methods and cutting-edge technology to pinpoint the exact culprit causing the infection.
So walk us through the process. What are the key steps involved in identifying H. influenza in a patient sample?
The first step is often a gram stain, a quick and easy way to visualize bacteria under the microscope. H. influenza will show up as those characteristic tiny gram-negative coccobacilli we talked about earlier.
That's our first clue. Their telltale appearance under the microscope.
But we need more than just a visual, right? We need to confirm their identity and figure out what they're capable of.
Exactly. That's where culture comes in. We take a sample,
Right.
whether it's from the respiratory tract blood or cerebrospinal fluid, and try to grow the bacteria on a special medium. And you mentioned earlier that H. influenza is a bit picky about its growth conditions. It's all about that chocolate agar, right?
You got it. Chocolate agar provides those essential X and V factors, hemin and NAD, that H. influenza needs to thrive. We also incubate the plates in an environment with increased carbon dioxide as that enhances their growth.
It's like creating a cozy little spa for these finicky bacteria.
That's one way to put it. And once we have the bacteria growing, we can perform a battery of tests to confirm its identity and determine its susceptibility to antibiotics.
antibiotic susceptibility testing is crucial for guiding treatment decisions,
Yeah.
especially in an era of increasing antibiotic resistance.
Absolutely. We need to know which antibiotics will effectively kill the specific strain we're dealing with, ensuring the patient receives the most appropriate therapy.
Now, I recall you mentioned a curious phenomenon called the satellite phenomenon. Can you shed some light on that for us?
Certainly. Some Haemophilus species, especially those that require the V factor, can only grow in close proximity to other bacteria that release this nutrient. Staphylococcus aureus, for example, generously releases NAD into its surroundings, creating a favorable environment for these fastidious Haemophilus species to flourish.
So it's like a bacterial buddy system, with one species supporting the growth of another.
Exactly. And visually, it's quite striking. You'll see these tiny colonies of Haemophilus huddled around a larger colony of S. aureus, like satellites orbiting a planet. It's a helpful clue in the lab, especially when we're trying to differentiate between different Haemophilus species.
I'm starting to see how the lab is like a detective's playground, full of clues and puzzles to solve.
It really is. And each test we perform adds another piece to the puzzle, helping us understand the nature of the infection and guide treatment decisions.
Now, I know there are various other diagnostic techniques available, both traditional and molecular. Can you give us a brief overview of some of the other tools in your arsenal?
Of course. Besides gram staining and culture, we also use biochemical tests to further characterize the bacteria. These tests assess the bacteria's ability to utilize specific sugars, produce certain enzymes, or exhibit other characteristic metabolic activities. It's like creating a metabolic fingerprint that helps us confirm the identity of the culprit.
So it's another layer of confirmation, ensuring we've got the right suspect.
Exactly. And in recent years, molecular techniques, particularly polymerase chain reaction, or PCR, have revolutionized the field of microbial diagnostics.
PCR has become the gold standard for many infectious diseases, hasn't it?
It has. It's incredibly sensitive and specific, allowing us to detect even tiny amounts of bacterial DNA in a patient's sample. This is particularly helpful in cases where traditional culture methods might be slow or challenging, such as with H. ducreyi, the causative agent of chancroid.
I'm realizing that the world of microbial diagnostics is constantly evolving, with new techniques emerging all the time.
You're absolutely right. And as technology advances, we're able to identify and characterize these bacteria with increasing speed and accuracy, leading to more timely and effective treatment decisions.
Speaking of treatment, let's move on to discuss the various strategies for managing Haemophilus infections. I'm sure our listeners are eager to learn about the latest weapons in our arsenal.
I'd be happy to delve into the therapeutic options, but first, it's crucial to emphasize the importance of prevention. And when it comes to Haemophilus, the Hib vaccine deserves a standing ovation.
The Hib vaccine has been a true game changer, hasn't it?
Absolutely. It's one of the most successful vaccines ever developed, dramatically reducing the incidence of invasive Hib disease worldwide. It's a testament to the power of immunization in protecting vulnerable populations from serious, life-threatening infections.
I'm curious, how exactly does this wonder vaccine work? What's the secret behind its success?
The Hib vaccine is a conjugate vaccine, meaning it combines the bacterial capsular polysaccharide, the key virulence factor we discussed earlier, with a protein carrier. This combination enhances the immune response, making the vaccine incredibly effective at inducing long-lasting immunity.
So it's like giving our immune system a cheat sheet, teaching it exactly what to target.
That's a great way to think about it. The vaccine essentially trains our immune system to recognize and attack the Hib capsule, preventing the bacteria from establishing a foothold and causing those invasive infections.
It's a beautiful example of how understanding the enemy's weaponry allows us to develop targeted and effective countermeasures. Now, aside from vaccination, are there any other preventive strategies for Haemophilus infections?
Yes, in certain situations, we might recommend antibiotic prophylaxis for close contacts of individuals with Hib disease.
Prophylaxis, what does that entail?
It involves giving a short course of antibiotics, typically rifampin, to individuals who have been in close contact with someone diagnosed with Hib disease. The goal is to eliminate the bacteria from their nose and throat, preventing them from spreading the infection to others or developing the disease themselves.
So is it a preemptive strike to break the chain of transmission?
Exactly. It's an important tool for controlling outbreaks and protecting vulnerable individuals.
Now, while prevention is always the ideal, we know that infections do occur. So let's dive into the treatment options for Haemophilus infections. I'm sure our listeners are eager to learn about the latest antibiotic strategies.
Absolutely. But before we delve into specific antibiotics, it's important to emphasize that treatment decisions are always individualized, taking into account factors such as the patient's age, overall health, the severity of the infection, and local patterns of antibiotic resistance.
That makes sense. A one-size-fits-all approach wouldn't be appropriate in such diverse clinical scenarios.
Exactly. Now, for invasive hive infections such as meningitis, we need to act swiftly and decisively. The antibiotics of choice are typically third-generation cephalosporins, like ceftriaxone or cefotaxime.
Why are these particular antibiotics preferred for such serious infections?
They have several key advantages. First, they're highly effective against H. influenza, including strains that might be resistant to other antibiotics. Second, they have excellent penetration into the cerebrospinal fluid, reaching therapeutic levels in the brain and spinal cord, where these infections can be particularly devastating.
So they're like precision-guided missiles, targeting the infection directly at its source.
Precisely. And finally, they have a favorable safety profile, which is crucial when treating vulnerable populations, especially young children.
Now, what about those less severe infections caused by non-capsulate H. influenza, like otitis media and sinusitis? I imagine the treatment approach is a bit different in those cases.
You're right. For uncomplicated NSIF infections, we often start with amoxicillin, a broad-spectrum penicillin antibiotic that's generally effective against these strains. However, if there's concern about resistance, or if the infection doesn't respond to amoxicillin, we might switch to amoxicillin clavulanate, a combination that includes a beta-lactamase inhibitor to overcome resistance mechanisms.
So it's all about tailoring the antibiotic choice to the specific situation, considering the severity of the infection, the patient's individual factors, and the local patterns of resistance.
Exactly. And it's crucial to remember that antibiotic stewardship plays a vital role in preserving the effectiveness of these valuable drugs. We need to use them judiciously only when necessary, and ensure patients complete the full course of therapy to minimize the emergence of resistance.
Antibiotic resistance is a global threat, and we all have a responsibility to use these precious resources wisely.
Absolutely. It's a collective effort involving healthcare providers, patients, and policymakers to ensure the future effectiveness of antibiotics.
Now, before we wrap up this fascinating deep dive, I want to touch on a topic that's become increasingly important in the management of Haemophilus infections: antibiotic resistance.
It's an issue we can't afford to ignore. H. influenza, like many bacteria, has developed various strategies to evade the effects of antibiotics, making treatment more challenging.
What are some of the specific mechanisms behind this resistance, and what are the implications for clinical practice?
One of the most common mechanisms is the production of beta-lactamases, enzymes that break down penicillin antibiotics like ampicillin. This is why amoxicillin clavulanate, which includes a beta-lactamase inhibitor, is often preferred over amoxicillin alone.
So it's like adding a bodyguard to protect the antibiotic from being destroyed.
That's a great analogy. Another mechanism is the alteration of penicillin-binding proteins, the bacterial targets that penicillin antibiotics bind to. These alterations prevent the antibiotics from effectively interfering with bacterial cell wall synthesis.
It's remarkable how these bacteria can evolve so rapidly to outsmart our best defenses.
It is indeed a constant arms race. And to complicate matters further, we're also seeing resistance to other classes of antibiotics, like macrolides, tetracyclines, and even fluoroquinolones, which were once considered our last line of defense.
So it's a reminder that we can't afford to be complacent. We need to stay ahead of the curve, constantly monitoring resistance patterns and developing new treatment strategies.
Absolutely. Continuous surveillance and research are crucial in this ongoing battle against antibiotic resistance.
Now, before we conclude this segment of our deep dive, let's turn our attention to the management of Haemophilus infections in specific clinical scenarios.
I'd be happy to discuss some practical considerations. Let's start with meningitis, one of the most feared complications of Haemophilus infection.
Meningitis is a serious and potentially life-threatening infection that requires prompt and aggressive treatment.
Exactly. In addition to antibiotics, we often employ supportive measures, such as intravenous fluids and medications to control fever and seizures. In some cases, corticosteroids might be used to reduce inflammation and prevent neurological complications.
So it's a multifaceted approach, addressing not only the infection itself, but also the potential consequences.
Precisely. We aim to not only eradicate the bacteria, but also minimize the long-term impact on the patient's health.
Now, what about those less severe NC-high infections, like otitis media? I understand that antibiotic use in these cases is sometimes debated.
You're right. There's ongoing discussion about the appropriate use of antibiotics for otitis media. While antibiotics are often prescribed, some studies suggest that they might not always be necessary, especially in mild cases.
So it's a decision that needs to be made on a case-by-case basis,
Hmm.
weighing the potential benefits against the risks of antibiotic use.
Exactly. Factors to consider include the child's age, the severity of symptoms, the presence of any complications, and the likelihood of bacterial resistance.
It's a reminder that clinical judgment and individualized care are essential in managing these infections. Now I think it's time to put our listeners' knowledge to the test. We've covered a lot of ground, and I'm sure they're eager to see how much they've retained.
I'm all for a challenge. Let's see how they fare.
All right, get those thinking caps on. Here's our first exam-style question. What are the 2 growth factors required by H. influenza for growth on artificial media? Take a moment to ponder that one. And we'll be back in part 2 of our deep dive to discuss the answer and explore its significance. Welcome back to our deep dive into the world of Haemophilus. Before the break, we left you with a challenging question. What are the 2 growth factors required by H. influenza for growth on artificial media?
Those 2 essential ingredients, as you may recall, are hemin, known as the X factor, and NAD, known as the V factor. Without these, H. influenza simply won't cooperate in the lab.
And this fastidiousness is directly tied to their unique metabolism, isn't it?
Precisely. Hemin is a crucial component of heme, a molecule found in hemoglobin that's essential for oxygen transport. H. influenza, unfortunately, can't synthesize heme on its own, so it has to scavenge it from its environment. Hence the need for the X factor.
So they're essentially relying on a hemin handout to carry out their respiratory processes.
Exactly. And then there's NAD, which plays a vital role in various metabolic reactions, particularly those involved in energy production. Again, H. influenza can't produce its own NAD, making it dependent on external sources, hence the need for the V factor.
It's fascinating how these tiny bacteria have such specific dietary needs. And this is precisely why we use chocolate agar to cultivate them in the lab, right? It's like providing them with a gourmet meal tailored to their unique palate.
You hit the nail on the head. Chocolate agar, with its rich supply of laced red blood cells, provides a readily available source of both X and V factors, making it the ideal growth medium for these finicky bacteria.
I'm starting to gain a real appreciation for the meticulous work that goes into culturing these organisms. Now, ready for our next brain teaser?
Absolutely, fire away.
All right, here it is. What is the primary virulence factor responsible for the invasiveness of H. influenza type B? Think carefully, and we'll be back shortly to dissect the answer.
Let's dive into the heart of Hib's invasiveness. The primary culprit, as you may recall, is its polysaccharide capsule, particularly the type B capsule. This structure acts as a formidable shield, allowing the bacteria to evade our immune system's watchful eye.
Remember, we touched upon the capsule earlier, but how exactly does it manage to outsmart our body's defenses?
The key lies in its composition. The type B capsule is made up of a unique arrangement of sugars called polyribosyl ribosyl phosphate, or PRP for short. This particular structure makes it incredibly slippery and difficult for our immune cells, like those valiant macrophages and neutrophils, to grab onto and destroy.
So it's like the capsule is coated in Teflon, allowing it to slip past our immune system's grasp.
Precisely. This ability to evade our immune system is what makes encapsulated strains, especially type B, so dangerous. They can multiply and spread rapidly throughout the body, leading to those invasive infections we discussed earlier.
It makes you appreciate the brilliance of the Hib conjugate vaccine even more, doesn't it? By exposing our immune system to this slippery PRP in a safe and controlled way, the vaccine teaches our bodies to recognize and attack it, effectively disarming those stealthy type B strains.
It's a remarkable example of how understanding the enemy's weaponry allows us to develop targeted and effective countermeasures.
Absolutely. Now, are you ready for another clinical challenge? This one requires you to put on your doctor's hat and visualize the patient.
Bring it on. I'm always up for a diagnostic puzzle.
All right, here's question number 3. Describe the clinical presentation of epiglottitis caused by H. influenza. Imagine you're examining a patient with this condition. What would you expect to see? We'll be back in a moment to discuss your observations.
Picture this: a young child, typically between 2 and 7 years old, is brought to the emergency room with a sudden onset of high fever, sore throat, and difficulty breathing. The child is anxious, struggling to breathe, with each inhale marked by a distinctive barking cough.
It sounds incredibly distressing for both the child and the parents. A true medical emergency unfolding before your eyes.
Absolutely. And as you examine the child further, you might notice drooling, a muffled voice, and an anxious, almost panicked expression as they struggle for air.
So the symptoms escalate rapidly and are very specific to airway obstruction. What's the key physical finding that would confirm your suspicion of epiglottitis?
If you carefully visualize the epiglottis using a tongue depressor, you'd likely see a cherry red, swollen and inflamed epiglottis. However, it's crucial to proceed with extreme caution.
Why is that? What's the danger in examining the epiglottis?
Even slight manipulation of the inflamed epiglottis could trigger a spasm, leading to complete airway closure and respiratory arrest. It's a delicate situation that requires a calm and measured approach.
So the priority is securing the airway, ensuring the child can breathe. I imagine that involves calling for an anesthesiologist and preparing for intubation.
Exactly. Time is of the essence in epiglottitis. Simultaneously, we would initiate treatment with intravenous antibiotics, targeting H. influenza.
It's a powerful reminder of how rapidly infections can escalate and how crucial prompt recognition and intervention are in saving lives. Ready for another brain teaser?
Absolutely. Lay it on me.
All right, here's question number 4.
Wow.
And it's all about treatment. What is the recommended first-line antibiotic for treating hive meningitis? We'll be back in a moment to reveal the answer and discuss why it's the top choice for this serious infection.
So we're facing a case of hive meningitis, a life-threatening infection that requires immediate action. We need an antibiotic that can not only effectively kill H. influenza, but also reach therapeutic levels in the cerebrospinal fluid, the fluid that bathes the brain and spinal cord.
And what antibiotic fits the bill in this critical situation?
The recommended first-line treatment for hive meningitis is a third-generation cephalosporin, such as ceftriaxone or cefotaxime.
And what makes these particular antibiotics the gold standard for this condition?
They have several advantages. First and foremost, they're highly effective against H. influenza, including strains that might be resistant to other antibiotics. Second, they exhibit excellent penetration into the cerebrospinal fluid, ensuring they reach the infection site and effectively target the bacteria.
So they're like those elite special forces units capable of infiltrating the most secure locations.
That's a great analogy. And lastly, they have a favorable safety profile, which is crucial when treating serious infections, especially in vulnerable populations like young children.
Now for our final question. Let's step back into the lab and revisit a powerful technique we touched upon earlier.
Intrigued to hear what you have in mind.
All right, here it is. What is the role of multilocus sequence typing, or MLST, in studying H. influenza? Think back to our earlier discussion about tracking these bacteria. We'll be back shortly to reveal the answer and discuss the implications of this powerful technique.
Let's delve into the fascinating world of multi-locus sequence typing, or MLST, a technique that has revolutionized our understanding of H. influenza by allowing us to explore its genetic makeup in unprecedented detail.
I remember it being described as a bacterial fingerprinting technique, but can you elaborate on how it actually works and why it's so valuable?
Imagine you're a detective trying to identify a suspect. You wouldn't rely on just one piece of evidence, right? You'd gather multiple clues, like fingerprints, DNA samples, and eyewitness accounts to build a solid case.
So it's like creating a comprehensive profile of the culprit, a molecular wanted poster, if you will.
That's a great way to put it. MLST works in a similar way. It involves sequencing specific genes from different parts of the H. influenza genome, creating a unique genetic fingerprint for each strain.
So each strain gets its own distinctive barcode based on its DNA. And by comparing these barcodes, we can trace their origins, track their movements, and understand their relationships.
Precisely. It's like having a global surveillance system for bacteria. This information is invaluable for understanding outbreaks, studying the emergence of antibiotic resistance, and developing effective control strategies.
It's a testament to the power of molecular techniques in unraveling the mysteries of the microbial world. And it highlights how understanding the enemy at a genetic level is crucial for developing effective strategies to combat them.
Absolutely. MLST has opened up a whole new world of possibilities for studying and managing bacterial infections.
Well, that concludes our 5 exam questions for today's deep dive into the fascinating world of Haemophilus species. We hope you found it challenging and enlightening. But before we sign off, I'd like to hear your final thoughts on what's most important for medical microbiologists to keep in mind when it comes to these bacteria.
I think it's crucial to remember that the microbial world is a dynamic and ever-evolving landscape. Bacteria like H. influenza are masters of adaptation, constantly finding new ways to survive and thrive.
So it's a reminder to stay vigilant, keep learning, and be prepared for new challenges that might emerge. We can't afford to rest on our laurels.
Exactly. Ongoing research, surveillance, and collaboration are crucial for staying ahead of the curve in the fight against infectious diseases.
Well said. Thank you again for sharing your expertise with us today. It's been a truly fascinating journey into the world of Haemophilus.
My pleasure. I always enjoy exploring the complexities of microbiology and sharing insights with fellow enthusiasts.
And to our listeners, thank you for joining us on this educational adventure. We hope you've gained valuable insights into the world of Haemophilus species and the importance of staying informed in this ever-changing field. Until next time, keep exploring, keep learning, and stay curious. We've delved deep into the world of Haemophilus species, uncovering their secrets and exploring the challenges they pose, from their microscopic characteristics to their remarkable ability to outsmart our defenses. It's clear these bacteria demand our attention and respect.
They certainly do, and as medical microbiologists, we're entrusted with the task of unraveling their mysteries and developing strategies to keep them in check.
Now, throughout our discussion, one theme has emerged repeatedly: the absolute importance of individualized care when it comes to managing Haemophilus infections.
Couldn't agree more. A one-size-fits-all approach simply doesn't cut it in the face of such diverse clinical presentations and the ever-evolving landscape of antibiotic resistance.
So as we prepare to wrap up this deep dive, I'd like to focus on the practical aspects of patient management. What are some of the key factors that you consider when making treatment recommendations for Haemophilus infections?
Well, first and foremost, we carefully assess the patient's individual circumstances, their age, their overall health, the severity of the infection, and any underlying medical conditions that might influence our approach.
So gathering a detailed patient history is absolutely crucial in guiding those decisions.
Absolutely. And then there's the ever-present challenge of antibiotic resistance. We need to stay abreast of local resistance patterns and ensure our antibiotic choices align with current susceptibility data.
It's like a constant game of chess.
Yeah.
Trying to anticipate the bacteria's next move and choose our weapons accordingly.
Precisely. And this often involves collaborating closely with other healthcare professionals, infectious disease specialists, pharmacists, to ensure the patient receives the most comprehensive and effective care.
It's a team effort, with each member bringing their unique expertise to the table.
Absolutely. And when it comes to managing these sometimes complex infections, that collaborative approach can truly make all the difference.
Well, I think we've covered a remarkable amount of ground today. We've explored the intricate world of Haemophilus species, delving into their biology, the remarkable adaptability, and the ever-present challenge of staying one step ahead of their evolutionary tricks.
I'd say we've given our listeners a solid foundation for understanding and managing these intriguing bacteria. But the journey doesn't end here. The field of microbiology is constantly evolving, and so too is our knowledge of these remarkable organisms.
So to our listeners, we encourage you to remain curious, to embrace lifelong learning, and to approach the microbial world with a sense of wonder. Because within those microscopic realms lie both challenges and incredible opportunities for discovery.
Beautifully put. And on that note, I'd like to extend my gratitude for the opportunity to share this deep dive with you and our listeners.
The pleasure was all mine. Thank you for lending your expertise and insights.
Yeah.
And to our listeners, thank you for joining us on this journey into the fascinating world of Haemophilus species. Until next time, stay curious, stay informed, and may your microbes always be friendly.